So it is evident from this particular diagram that MREB protein is, which is an actin homolog in prokaryotes, is required for maintaining the cell shape, and it is associated with some other proteins, MBL. You need to remember this. Now, other protein, for example, crescentin, which is an intermediate filament homolog in eukaryotes. So in prokaryote, it is having some role. For example, crescentin is shown here. So this blue color protein, which is shown here, is crescentin. So what is the function of crescentin? Crescentin is required when cell is bending from one side, so either from here or from this side. So this is a role of crescentin protein. So like, for example, this is intermediate filament homolog. Crescentin polymerizes along the length of inner curvature of the cell, so help the cell to bend or maintain this kind of structures, for example, in vibrio cells or coma-like cells. Cells depleted of crescentin are a rod shape. So suppose if some cell do not have this protein at all, this crescentin is depleted from here. So what can happen? Suppose this is not present. So let me put the crescentin is not present. Fine. So what will happen? This cell will not bend from this particular location. So the cell will be purely the rod shape in nature. So only because the crescentin is present, therefore cell is having some kind of curvature, or some, for example, spiral cells or these vibrio cells. These are specifically having this crescentin protein. Also, then there is a protein FTS-Z. This is remaining. So let me show you the table first. So this FTS-Z is actually the tubulin homolog, which is required during the cell division and widely observed in the bacteria and archaea. So this FTS-Z, now what is the function of FTS-Z? Let's look at the diagram. So this FTS-Z is making a coil-like structure at the center of the cell. So, and also this MREB protein, which is actually present as a coil in along the length of bacterial cell, it will also become localized at one place when it comes to the division. When bacterial cell want to divide, generally bacteria are divided every 20 minutes or so, 20 or 30 minutes. So each time when the bacterial cell divide, they will come at a common location. So confirming that the two daughter cells which are forming after the cell division are equal in size. Okay, so this is the main function of FTS-Z. So I will also show you how this will happen actually. So I am also having one more slide. Okay, so here. Sorry. Okay, this we have discussed. This. No, not this. Okay, this one. FTS-Z actually shown. Oh, sorry. Better here. Why this is not fine? Okay, so let me zoom in. Fine. So there are two protein. Actually, this MREB protein, MREB protein is actually for murin cluster B. And we will discuss FTS protein, FTS-Z also. So it is given here because this Z is for Z ring. Okay, so tubulin homolog found in most of the bacteria and archaea, forming the Z ring, like tubulin polymerizes to form the filaments and so on. Okay, so here is the main theme. There are two proteins which are present. So one protein is MinCD is also expressing at the same time. So one is this we were discussing, MREB protein. One was FTS-Z protein. So main function during the division is of FTS-Z. Okay. And now what is the function of this new protein? MinCD, actually I did not discuss MinCD with you, but actually it is given in the table. Actually, if you find here, this is Min family. So MinD is forming a complex with MINC, and this is expressed inside bacterial cell, ensuring that because this ring, this called Z ring, so this FTS Z ring should not form towards the poles. Suppose the FTS Z ring form at the pole of the bacterial cell, rod cells. So what will happen? What can happen? One cell after division will be small. One cell, which is the daughter cell, actually will be large in size. So to ensure that both the cells are of equal size, so this min CD is uh, holding the FTS jet in the center, so that the polymerization only take place at the central position of the parent of the mother cell. So the daughter cells which are formed after division are of equal size. So suppose you are able to understand this, because this is very clear from this particular diagram. This min CD is a kind of protein. So it is very similar in the structure to MREB protein, but it is having some different function. MREB is having function, it is supporting, or it is giving some kind of shape to the bacterial cell, because it is polymerizes according to the length of bacterial cell. But min CD is polymerizing to stop the polymerization of FTS jet towards the poles. So you can see here they are maintaining FTS jet in the center, so that a ring-like structure will form. And finally, when the bacterial cell will divide into two, as shown here, so because these are dividing from here, and this knot, you can see these depressions here. So these depressions in the center are due to that jet ring, which is made up of FTS jet. Okay, so finally the cell will divide from the center, and two equal sized daughter cells will be resulted. Now, what is actually required? Okay, so one thing, bacteria ensure that both the daughter cells which are formed after division should be of equal size. Second thing, bacteria need to circulate all the plasmids in equal number in both the cells. Suppose bacteria is having one plasmid. So first of all, that particular plasmid will divide so that the two plasmid will form before division. Then you can see the bacterial cell size will be increasing. So because the cell is going to divide, and up to division it will increase in size, as well as it will divide its plasmid and DNA. Okay, so you can see this thread-like protein. So this is also having some function, PAR-M protein. Okay, so and there is a complete family of PAR proteins. So this PAR protein is actually required for partition. Okay, so it is made up. It is having three proteins which are isoforms, you can say. So M, C, and R. If you look in the table, PAR. So PAR A is given here. So here are some other PAR proteins are also given in this particular diagram. Now, what is the function of this another protein, PAR M protein and PAR C, and all these are proteins. So first of all, so bacterial cell want to give equal number of plasmid to each daughter cell. Okay, suppose if there is only one plasmid and bacterial cell divide, so either one cell will receive the plasmid and other cell will not able to receive the plasmid at all. So uh, it is a kind of partiality. Okay, so bacterial cell want to give equal number of plasmids to both the daughter cells. So what can it do? First of all, it need to divide so that equal number of plasmid can be distributed to each daughter cells. And then these plasmids should move towards the poles, because it is ensuring, the FTS jet is ensuring that bacterial cell is dividing in the center only, so that the bacterial mother cell need to push these two plasmids towards the opposite poles, because bacteria know that the poles will ultimately forming a daughter cell. Okay, and it is going to divide from the center. So this protein, which is actually moving or pushing these two plasmids towards the opposite poles, it is made up of two different proteins. There is one red colored dot here and there is one black thread here, if you're able to differentiate. So there are two proteins. One is PAR-M. This is not black, actually, this is green. So green filament is anchored to PAR-C and PAR-R, which are shown in the form of red dots. So the red dots are actually made up of two proteins, C and R. CR, you can say, it is a dimer. And M protein is this green or black, whatever you are able to see, this filament which is joining these two red dots. Okay, so when this polymerizes, so what will it do? It will push both the plasmids towards the opposite poles, so that when bacterial cell will divide from the center with the help of FTSZ protein, or a combination of, so as you see from, where was that particular diagram? So hope this is clear. And then this par M, which is a green filament, will elongate, thereby pushing each plasmid to the opposite pole of the dividing cell, ensuring that each cell will able to receive equal number of plasmids. It is shown here also. So this green filament is actually par M, and these dots are actually having two proteins. One is C, one is R. Okay, and then finally, okay, so this one actually, I was looking this. So finally, these two proteins will accumulate in the center, make a septum-like structure, and the bacterial cell will make these curvatures, or you can say these depressions in the center, and finally will divide into two. Fine. If you have any question from this particular discussion, actually I'm discussing this particular portion from the previous class, because these proteins, okay, so it is from the Prescott. Page numbers are also given around 159, 160. All these diagrams are given. I'm having Prescott fifth edition, sorry, eighth edition actually. If you have Prescott, please go through these page numbers. You will finally find these diagrams. Okay. Also, so we have discussed this. This we have discussed. If you have any query from these diagrams, whatever we have discussed, because we have discussed so many proteins, MIN, CD, FTSZ, MREB, MAMK protein, and all these, it is hard to remember them. But actually, with the help of this table only, you will able to differentiate them, as well as you are able to answer some of the questions, very tricky questions. So you need to remember their homologs in eukaryotes, because question will come like this only, or their function related questions may be asked. Okay, so there is a query actually. So someone, how does filament degrades? Okay, so it is a polymer of means amino acids. Obviously, it is a protein. So the similar mechanism, bacteria need to degrade it in a similar way it is having for degradation of other proteins. Okay, so proteasomal degradation. I read MINCD protein is, yes, this is correct. Yeah, MinCD protein is actually an inhibitor of Z ring assembly. Inhibitor in the sense it will not inhibit the formation of Z ring. It will form anyhow, because if the FTS Z ring will not form, bacterial cell cannot divide at all. But it will inhibit the formation at the poles. So, okay, so let me read some text so that you are able to understand the picture in more detail. Okay, so okay, Priyansh is also having a query. So how this FTS A is different from FTS Z? Okay, fine. So we will go through the text. So I was thinking like I will finish it quite early, but no hurry. We will discuss in more details. Fine. So first of all, you are able to understand the function of MREB protein. This is a full form for murin cluster B. You can note this down. In the rod-shaped microorganisms, in coccus, this is absent. MREB protein is similar to the eukaryotic actin and polymerizes to form a spiral around the inside of periphery. So it will run along the length of rod-shaped like this bacteria. Okay. So first of all, Niharika has asking one question here. FTS Z assemble in the center of the cell to form a Z ring. Let me zoom in. So FTS-Z is a full form for fermenting temperature sensitive mutant Z. And okay, so Z and A are two different mutants. If Z is not forming, the bacterial cell will not divide. So it is a kind of protein which is helping bacteria to divide into two equal daughter cells. Okay, so first query, let me read this line. FTS-Z assemble in the center of the cell to form a Z ring, which is essential for the separation of cell, essential for separation, okay. Min CD together with other min proteins oscillates from the pole to pole. So it is oscillating motion. It is having some kind of oscillative motion. And you see oscillations will help FTS-Z to only polymerize in the center. It will not allow FTS-Z to polymerize at either poles. It is having some kind of oscillations or oscillatory motion. Okay, so you can try this when you are having a big vessel or a container and it is filled with water. With your hand, if you try to oscillate or will rotate your hand, and there is a loop-like structure will form in the center. Fine. So similar thing is actually happening here. When is it is oscillating, it will not allow the polymerization of FTS-Z at the poles, so that in the center only this protein can polymerize. So this is first query and answer to the first query. So from pole to pole, thereby preventing the formation of an off-center Z ring. Off-center Z ring means only the ring can form in the center. It cannot form beyond or towards the poles. Fine. There is one more query. Let me see what is this. Okay. So this is one. Second is so each cell, Trisha, having some degradation mechanism for protein or polypeptides when they are of no use or the function is over. So like other proteins which are normal in bacteria, they also degrade these polypeptides of proteins. Okay. And the third query is actually these are two different mutants. One is mutant Z, one is mutant A. And also there are differences in FTS-Z and A because FTS-A is actually actin homolog, whereas tubulin homolog is FTS-Z, which is making this Z ring. But actually both are similar, both are having similar functions. So if someone is asking me differences, I'm telling one is tubulin homolog, one is actin homolog, both are different mutants. But actually they are recognized from different mutants, but actually they are found to be having the similar function. They are playing role in cell division. So okay, fine. Hope this is very understandable. We have discussed in our last class also, but actually the core thing, I want to show you some diagrams so that you are better able to understand. It will create a picture in your mind so that you will be able to remind where we have studied these particular proteins and what may be their possible functions. Fine. Rest, actually, I expect from you that you will remember these homologs at least. So, tubulin homologs are FTS-Z. Actin homolog, it is itself A, so you need not remember. MAMK protein and MREB protein, which are actually, it is required magnetosome, and MREB protein, which is actually required for maintaining the cell shape or segregating the chromosome. It is actually actin homolog, whereas crescentin protein, which is absent from the rod-like bacteria, and it is actually required for some curvature or some curve-like bacteria will have this particular protein. So as shown here, so this blue color protein is actually crescentin. So only when rod shape of the shell will distort, so only then they will be expressed. Otherwise, they will be absent from rod-like bacteria. Okay, so anything missed, let me look into the text. If anything is very important, I will let you know. Okay, so okay, so par proteins we have discussed. Okay, so this par protein is for par for partition. It is written here, and M for motor. This par is having very similar to MREB protein is an actin homolog, and it polymerizes in an ATP dependent way to form a long filament. So let me clear one thing first. So here, when you see this par A is actually written like unique bacterial cytoskeletal protein, but here par M protein is in the reference book in the Prescott, they have discussed this. This is a actin homolog actually. So please, in this table, if you are writing or noting this particular table, please add RM protein here somewhere. Okay, so that you will remember this is an actin homolog. Okay, Niharika is having a query. You mean the cellular concentration of FTSZ regulate the frequency of division? Okay, so you can correlate this like this. Okay, so not cellular concentration. It may be present in the cell in any concentration. It does not matter actually, but the polymerization will affect the cell division, and this will only polymerize when cell need to divide. This is not like if it is having more FTS jet, so cell will divide each 5 minutes or 10 minutes or like that. There is some different reason why bacterial cell divide every 20 minutes. I will discuss that also in this particular class. But okay, so this is different thing. But yes, if it polymerizes, then finally the cell will divide. So okay, so this is not the text actually for today's class. We need to discuss this from this PDF. First of all, so I was telling like you need to add par-M in the actin homolog class. It polymerizes in an ATP dependent way to form a long filament within the cell. Then there is some R represent for a repressor and C for a centromere like. We have discussed R and C, which are making some red dot like structures in the next page. They are given. So this is par, C, and R. R for repressor, C for centromere like something. Bind to the origin of replication sequence of each R1 plasmid. Okay, so someone can ask me this particular question. So actually, I have answered this. How this R protein and C proteins, which is a centromere-like protein, are able to bind to the plasmid specifically, so that okay, you understand what I'm saying? So see here, these red dots are able to bind to the two different plasmids. What kind of interactions they make? This may be a question. Actually, they recognize the origin of replication sequences in these plasmids, and therefore, when this green filament or this par M protein will polymerize, which is for M4. Uh, sorry, I forget what was M. So M for motor protein. Okay, so this motor protein is actually required. The polymerization is required so that its length will it will elongate and push the plasmids towards the opposite poles in the same cell. So interactions are actually in between the proteins, and it will recognize the origin of replication sequence of each R1 plasmid and link the motor protein or partition protein to the plasmid. Once the three proteins are attached to the plasmid, both ends of para filament elongate, thereby moving each plasmid to the opposite ends or the poles of the same cell. Clear? So then, Okay, so it is also written here in E. coli, there is a complete complex. This is called min-CDE complex, which is made up of three different min proteins, limits the Z ring formation to the center of the cell. Please note this. So certain things we have discussed actually. So what you need to remember from this, one is this is made up of three different proteins, and second thing is this Z ring is a homolog of tubulin. I told you already. Uh, then everything is fine. So everything other is actually discussed. So what is the role of FTS chat? So it will develop some kind of constriction or from where the cell will going to divide actually so that a septum can be formed. So this septum formation is a function of FTS jet. Then we move to the next page. Okay, so you may have one question, how bacterial cell is able to divide every 20 minutes, how it is whereas actually the so please read this out, you will get your answer. The preceding discussion of cell cycle described it occurs slowly growing E. coli cells. In these cells, the cell cycle take approximately 60 minutes. So some points are important. I want you to please note them. Each cell at least generally requires 60 minutes, but bacterial cells are able to divide every 20 minutes. So how this division, fast division is achieved? So despite the fact that DNA replication actually require 40 minutes and the bacterial division require 20 minutes. So overall 40 plus 20, it is requiring 60 minutes. But it is not the case. They are able to replicate or able to divide actually every 20 minutes. So they are doing some kind of extra work. So what is this extra work? E. coli accomplishes this by beginning a second round of DNA replication and sometimes even third or fourth round. So minimally it will second or third or in some cases actually which are very fast dividing bacteria, they can go up to fourth round of DNA replication. So suppose if the bacterial cell is uh, able to start the DNA replication before it is going to divide. So before the first round of replication is completed. Thus the progeny cells receive two or more replication forks already. So therefore, and the replication is continuous because the cell are always copying their DNA. So whether they are dividing, whether they are not dividing means for 20 minutes. So they are continuously making or replicating their DNA. They are they are actually in the second round of DNA replication, or in some cases they are in third or fourth rounds. And when the cell divide, they already receive two or more replication forks. Therefore, with the help of all this, the bacteria can divide every 20 minutes. Fine. So, okay, so one more information is here actually. So once the Z ring forms, the rest of division machinery, sometimes called the divisome. So if someone asks you what is divisome, so actually this is the division machinery with the help of which bacteria is able to divide. And now you are familiar that there are some proteins which are homologous to either actin, tubulin, or intermediate filaments. So this machinery is called divisome, is constructed as shown in above diagrams. Okay, so first one or more anchoring protein link the jet ring to the cell membrane. So this jet ring is made up of FTS jet and some other protein link this particular protein or this jet ring to the cell membrane. Then cell wall synthesizing machinery is assembled. And although numerous components of the division machinery have been identified, the function of many are still unknown, as we have discussed from the table. So some proteins, even today, we do not know their function. For example, this protein, which is a tubulin homolog, B tubulin A and B tubulin B, which is made up of two subunits A and B, function is largely unknown, and actually it is required for horizontal gene transfer. Like this, we do not know about the function of some of the proteins or the proteins of this machinery. But these invaginations are actually accompanied by this jet ring of the cell membrane, and synthesis of the septa wall or the septum formation is completely dependent upon the formation of jet ring or FTS jet. This is clearly written from in the text, and you can read this from here. Fine. Then, okay, so this can be a point also. So one protein, because this is complex, is made up of three different proteins, C, D, and E protein. Mainly this Min-C is an inhibitor, so it will block the septum formation. You know, septum is made up of FTS jet. It will block the septum formation. It oscillates with min B, and since concentration of min C is very, very high at the poles, septum formation is forced to occur at the center of the cell only. Okay, so if this is a query, so out of this complex, one is a main inhibitor of this FTS jet septum formation, and that is min C actually. And because the concentration of min C is high at the poles, it will not allow at all the formation of FTS jet ring or jet ring at the poles. Therefore, there is only chance of formation of FTS jet ring in the center of the cell, like this. Then, okay, so we have discussed all these proteins almost, which are given in this particular table, but there are some other proteins also which are not given in this particular table, and they are also reported, and they are part of this divisome proteins complex, which is a complex machinery required for the division of bacterial cell into two daughter cells. So, for example, this FTSA we have not discussed, GIP-A protein. So, these FTS Z is having role in the formation of Z ring, whereas FTSA and this ZAF, GIP-A or JAP-A, whatever you call it, it is an anchor Z ring to the plasma membrane. So, this is that particular protein we have discussed at last page. So there is one protein which is actually required so that this Z ring can be attached to the plasma membrane. So this FTSA and this other protein GIP or JAP-A is actually anchoring this Z ring to the plasma membrane, so that these so very clear in this particular diagram, if you are able to visualize, there is invaginations are formed at the both sides of these cells. So when this Z ring, which is not drawn here, let me draw the Z ring for you. So suppose this is the Z ring, and sorry, this is not the good color. Actually, I need this. So let me fine. So this is Z ring. Okay, so this is Z ring. Also, there is a protein. So let me draw one more protein. So suppose this we take and we have this in hand. So this is another protein on both the poles. Till now, we are able to understand that this is FTS jet ring made up of FTS jet, and this is having two proteins. One is FTSA, and the second is actually what we call it, JIP or JAPA. Okay, so this is not very clear here, but actually there are two proteins on both the sides which are anchoring this jet ring to the plasma membrane. Now, when this will pull this, or it will make a septa or septum-like structure, it will pull both the plasma membranes from the center so that a pressure will be created and this cell finally divide into the two daughter cells. Fine. So that's all. All right, we have discussed this. Then there are some other protein, for example, FTS, E, and X. We do not know what are their functions, as I already told you, and we are unable to find their functions yet. FTSK, so this is another FTS member. This K is actually chromosome segregation and separation of chromosome into the dimers. Not given in this table actually. Okay, so no worries. You need to remember chromosome segregation is a function of FTSK. So during the separation of chromosome dimers, it is required. And there is another FTS member that is a makeup of this QLB. This is a complex and we do not know the function. FTS I and W are required for peptidoglycan biosynthesis. We have discussed peptidoglycan synthesis in the bacterial cells. So these two proteins are also involved in the formation of peptidoglycan polymers. Okay. Then there is this AMI-C and ENV-C, which is having respective roles. So first one is amidase and second one is a hydrolase activity. So they are also involved. So this is amidase. Amidase in the sense it will break the amide bonds or peptide bonds. So it may be a kind of protein which is, as Trishla asked, which can be required to degrade the polypeptides, okay, and hydrolase activity. So these are also having role in the formation of some other protein activities. Okay, so the second topic of this particular class is the flagellar motor. So first of all, let's start from the structure we have already discussed. Okay, so I told you based upon flagella, the bacterial cells can be monotrichous, amphitrichous, lophotrichous, or peritrichous. I have to see this particular diagram. So based upon flagella, you can name the bacterial cells. If they do not have flagella at all, they are called atrichous. We are moving sequentially on, Krishna. What is, you are not able to understand actually. So till here, actually, we have discussed cytoskeletal proteins in bacteria. Now we are moving to next topic. So if you look into your book in the Pathfinder, so let me show you. Actually, we are moving very sequentially. There is no problem at all. So for the today's class, I have scanned this from the Pathfinder. So we have discussed this much in our previous classes. Protoplast and spheroplast we have covered. Then outer membrane also I told you in one class. So outer membrane structure we have discussed with the help of this reference diagram. So this is for gram-negative envelope, which is having a plasma membrane and outer membrane. This also we have discussed. I also told you that they are having some brown lipoproteins, which are kind of connecting links between outer membrane and peptidoglycan cell wall in gram-positive bacteria. Okay. I will discuss this also in detail. Actually, I will finish all the theory which is given in the Pathfinder. Each line is very, very important. Then the next is actually we need to discuss capsule and slime layer or your whatever is present outside the cell wall, outer membrane or cell wall. So capsule may be present in both gram-positive as well as in gram-negative bacteria. Then there are some structural, for example, plasma membrane, cytoplasm, and these things you know actually very well. Yes, I missed this portion, so I will come to these stored granules. For example, magnetosomes we have discussed. We need to discuss gas vesicles, polyphosphate granules, carboxysomes, and glycogen or some PHP granules, for example. This portion we will discuss abhi thodi der mein. And I'm directly coming to this flagella actually because I need to cover this portion also. Okay, so don't worry. Actually, whatever sequence is given in your Pathfinder, I will not skip that particular sequence. We will move sequentially. Only thing is I will add some extra things from the Prescott, which should be done because very little things are discussed when it comes to some molecular structure and like this in Pathfinder. So whenever required, we will move to Prescott. Otherwise, we will move according to the sequence which is given in your book Pathfinder. Okay, so actually I have discussed this particular structure in previous class. Therefore, I directly came to this particular structure. So depending upon flagella, you can name the bacterial cell. If it is not having a flagella-like structure, it can be atrichous. Okay, so I think it is given here also. So number and distribution of flagella. Number and distribution of flagella on bacterial surface are characteristic for a given species, and hence they are useful in identifying or classifying the bacteria. Okay. If they are having only single flagella, so first of all, you should know the function of flagella. Okay. So as we know, flagella are generally required for the movement in bacterial cells. So locomotion is main function, but this is not true 100% because in some cases it is also found that this flagella-like structure can be used for attachment to the surfaces or some other functions. Okay, so I will show you that portion also. This is monotrichous. If the bacterial cell is having flagella over both the ends, this is amphitrichous. If they are having a bunch of flagella on one pole of bacterial cell, this is lophotrichous. And if the flagella are present all over the bacterial cell, this is peritrichous. So you can get all the things from here. Cluster of flagella, lopho. Peritrichous, peri means if the flagella are around, means all around the bacterial cell, the flagella are present. So they are given here. Now I will move back. So let's start from this thing we have left. Okay, so okay, no problem. So we will discuss. There is no doubt we have discussed protoplast and spheroplast in our previous classes. These protoplast and spheroplast can be formed from normal bacteria, either from gram-positive bacteria or from gram-negative bacteria. If you treat those particular gram-positive and gram-negative bacteria with penicillin antibiotic, you will get protoplast or spheroplast. So what is the difference? The plasma membrane and everything within plasma membrane is actually protoplast. So what is the normal structure of gram-positive bacteria? So gram-positive bacteria is having a plasma membrane and it is covered with a thick peptidoglycan cell wall, which is generally 20 to 80 nanometer thick. So this is called protoplast. Suppose you inhibit the cell wall formation. So protoplast actually do not have cell wall. Similarly, in gram-positive bacteria, if they are incubated with an antibiotic penicillin, the cell wall formation will not take place, as we had discussed the cell wall formation or peptidoglycan synthesis. So penicillin is an inhibitor. And in isotonic solution, they will make protoplast because the cell wall formation will not take place in the presence of penicillin. So gram-positive bacteria will give you protoplast, whereas if this penicillin inhibits the peptidoglycan synthesis, it is written here. Gram-negative bacterial cells, if they are grown in presence of penicillin, what can happen? These gram-negative cells retain their outer membrane after penicillin treatment and are classified as spheroplast because some of their cell wall will remain. So because this outer cell wall, so first of all, I will show you what is the structure of gram-negative cells. So this is gram-negative envelope. Here, what is additional, what is in addition? So as I told you, these gram-negative bacteria are having two membranes. One is plasma membrane and one is outer membrane. So this outer membrane is not inhibited in presence of penicillin. If you grow these cells in presence of penicillin, outer membrane will remain. Also, some peptidoglycan cell wall, though it is very thin in gram-negative bacteria, but some portion will remain because it will protect the cell wall. Okay. So because at least they have this outer membrane along with this plasma membrane, and there is little peptidoglycan cell wall. So these structures are called spheroplast. So what you need to understand from this text, if you grow gram-positive bacteria in presence of penicillin, you will get protoplast. If you grow gram-negative bacteria in presence of penicillin, you will get spheroplast. And you need to grow these cells or treat these cells in isotonic solution. Otherwise, what will happen? Either your bacteria will swell or your bacteria will shrink. Okay, so then we move to outer membrane. We need to discuss outer membrane in detail, although I told you that outer membrane is made up of three major components. One is lipopolysaccharide layer. So this LPS is made up of O antigen, lipid A, and core polysaccharide. Core polysaccharide and antigen. So let me show you the diagram so that you can better understand this. So this is outer membrane. Here the red portions which are looking at the outer membrane are actually lipopolysaccharides. Why they are lipopolysaccharides? Because they are sugars. These fiber-like structures which are at the top are sugars, polysaccharides, which are attached to the lipid portion in outer membrane. Therefore, they are called lipopolysaccharides. So this outer membrane is mainly made up of lipopolysaccharides. The second thing I told you that this outer membrane is more permeable than plasma membrane because it is having porin-like structures which will allow the low molecular weight compounds to pass across the outer membrane. Yes, outer membrane actually I discussed in previous class, but not in detail, not from the Pathfinder, only from this book, from this page only. So no problem at all. Let's discuss it again so that you are able to better understand this. Okay. So whenever it come to gram-negative bacteria, they are more harmful, more virulent, and their virulence is actually due to presence of this lipopolysaccharide layer, or you call it outer membrane. So because they are having this outer membrane structure, they can be protected, well protected from several antibiotics, and therefore they are more pathogenic or more harmful. One more reason for gram-negative bacteria, why they are harmful, because they can secrete endotoxins. What are these endotoxins now? Because this LPS layer is made up of three components. What are these three components? These three components are core polysaccharide. So as discussed here also, these are three uh, components of LPS, or this outer membrane. One is called lipid A, one is called core polysaccharide, and one is called O antigen or A antigen, whatever you call it. So this O antigen is mainly these fiber-like structures which can be seen at the surface of the cell, which are made up of polysaccharides. Okay, so these fiber-like structures are required whenever there is some immune response against gram-negative strain or bacteria. So these antigens are recognized, therefore they are called O antigens. But what is the problematic portion in this gram-negative bacteria? Why they are so much harmful? Because they are having this lipid A portion, and this lipid A portion is actually toxic. So this is toxic. And since this lipid A portion is toxic, several gram-negative strains are actually said to secrete endotoxins. This endotoxin is nothing but this lipid A portion which is present in the lipid polysaccharide layer or in the outer membrane. So let's discuss this and finish this off. Okay, so in addition to the cell wall, gram-negative bacteria contain an additional membrane. You need to underline this because this is very important. Okay, so this additional membrane is actually making these gram-negative bacteria more pathogenic, or you can say these gram-negative bacteria are having two membranes. This additional membrane is nothing but the outer membrane or the LPS membrane. It is also called LPS layer. Constituents of this layer are not completely similar to the plasma membrane. It contains lipopolysaccharide, LPS, lipoproteins, proteins, and phospholipids. There are certain differences in molecular makeup when you compare the plasma membrane with an outer membrane. So if you are not able to, still are not able to get the message, you can see this particular structure. So see here, the plasma membrane is made up of lipid bilayer and the amided proteins. These proteins can be peripheral proteins or integrated proteins. Okay, and purely these are phospholipids which are making the plasma membrane. But if you look the structure, if you're looking at the structure of outer membrane, so it is having these lipids. Also, you will see that here they have drawn them with the yellow color. Here they have drawn them with some red color. Okay, so there is obviously some structural difference between outer membrane and plasma membrane. So what are these actually? So they are attached to sugars or carbohydrates. Therefore, they are called lipopolysaccharides. Whereas here they are not attached to any sugar moieties generally. Okay, so what is the advantage of addition of these sugar moieties? So these will act like antigens. They can be recognized by the immune system of our body. Also, you will look at the molecular makeup. They are having some porin-like structures. So these green channels which are present in the outer membrane are actually required to transport low molecular weight molecules across the outer membrane, whereas plasma membrane do not allow these molecules or some hydrophilic molecules to move across the plasma membrane. So in fact, the outer membrane is more porous due to presence of these pores or channels. They will allow the movement of some of the hydrophilic molecules, low molecular weight molecules. Another thing is these Brown's lipoproteins, which are present in gram-negative bacteria. So these are certain proteins which are connecting your cell wall or peptidoglycan structure with the outer membrane. So these are called Brown's lipoproteins. Now, the lipid and polysaccharides are linked in outer membrane to make an lipopolysaccharide or LPS layer. Because of this, the outer membrane is often called LPS layer. LPS consists of three components. We have discussed core polysaccharides, O polysaccharide is called O antigen or O side chain, and lipid portion is actually lipid A. Now, this is a direct question. This has been asked in previous years. This lipid A is the main culprit, which is giving the pathogenicity to the gram-negative bacteria actually. So this lipid A is not a glycerol lipid actually. Why this is toxic? Because this is not a glycerol lipid, which is normally present in our body also. Uh, it contain, it is something different. So it contain two glucosamine sugar derivatives. If you look the structure, looking at the structure here, so there are two derivatives of glucose molecule. These are called N-acetyl glucosamine, and these are linked to either pyrophosphate or phosphate. Here they are shown that the phosphate is attached, but these two sugar derivatives are N-acetyl glucosamine sugar derivatives, each with fatty acids. So there are some fatty acids are connected. So the lipid A portion is actually made up of three further components. One is phosphate, one is some sugar derivative, for example, N-acetyl glucosamine, and some fatty acids. And the fatty acids and the phosphate or pyrophosphate, which is attached to the sugar derivative. Lipid A is often toxic, need to underline toxic. As a result of this, the LPS can act as an endotoxin. O-polysaccharide function as an antigen, and it is useful for distinguishing the species of gram-negative bacteria. If you want to classify gram-negative bacteria, I want to distinguish one species from the other or some strains. So with the help of this O-polysaccharide, we can differentiate them. Otherwise, everything will remain almost similar. Now, actually, I have missed this. So we know that certain so this is the rule of the nature, that the sugars are generally D sugars and the amino acids are generally L amino acids, which are present in animals or in the life forms. But bacteria are an exception, since the bacterial cell wall is having some D amino acids, as well as its the gram-negative bacterial outer membrane, which is a lipopolysaccharide layer, may have some these L sugars. For example, L-rhamnose. So, for example, this KDO. What is KDO? Ketodeoxyoctonate. Man is something, mannose. Glucose, N is N-acetyl glucosamine. P for phosphate or pyrophosphate. And RHA, it is written here. RHA is actually rhamnose, L-rhamnose. So, in bacteria, L sugars or D amino acids can be found. So this core polysaccharide is made up of these subunits. So it is written here, everything is written, and their abbreviations are also given in the text. This we have already discussed, as these are having pore-like structures, so they will allow the low molecular weight substances to pass across the outer membrane. Therefore, it is allowing hydrophilic molecules, so it will be more porous in nature. Okay, so the next question is, what is periplasmic space? If you are able to understand this diagram in gram-negative cell envelope, so okay, let me annotate this so that you can better understand. So sorry, this is not visible actually. So if you can see this, the difference between the gap between the plasma membrane and an outer membrane. So this gap is actually called periplasmic space. So what should be noted here, in gram-negative bacteria, because gram-negative bacteria is having an outer membrane and it is having this space is very large in case of gram-negative bacteria. So periplasmic space is more in gram-negative bacteria, whereas gram-positive bacteria is actually having less space between the plasma membrane and the cell wall. So in gram-positive bacteria, this is only this much. Okay, so this much is present in gram-positive bacteria because gram-positive bacteria do not have this outer membrane. So first thing is gram-negative bacteria is having more periplasmic space. The second thing is this periplasmic space is filled with some solvent that is called periplasm. So let's discuss this in detail. So the gap or the space between plasma membrane and the outer membrane gram-negative bacteria is called periplasmic space. Substance which is present in the space is periplasm actually. A smaller but a thinner or small periplasmic space is present or found in the plasma membrane, between the plasma membrane and the cell wall in gram-positive bacteria. So what is the overall take-home message is gram-positive bacteria is having small or little or thinner periplasmic space, whereas gram-negative bacteria are having more pronounced periplasmic space. So periplasm content will be more in gram-negative bacteria. Then there is a layer or a substance which is present outside, either outside the outer membrane or outside the cell wall in gram-positive bacteria or in gram-negative bacteria. So because gram-positive bacteria is having plasma membrane and a cell wall, so the periplasmic space is small. You can see from this diagram also. They are having some capsule-like structure outside the cell wall, whereas the periplasmic space is more in the gram-negative bacteria because this is a space between outer membrane and plasma membrane. And the cell wall is actually present in the periplasmic space. So this is also one information you need to remember. So cell wall is present in periplasmic space in gram-negative bacteria. Now, what is capsule? So capsule is also called slime layer. Actually, depending upon if it is hard, it is capsule. If it is something loose or deformed, then it is called slime layer. Both are same thing, only difference in their texture or molecular makeup. So glycocalyx, actually capsule and slime layer both are actually called glycocalyx. So this is thick, high molecular weight secretory substance which is actually secreted by these cells and deposited outside the cell so that can protect the cell against host defenses or so on. And is present in many bacteria external to the cell wall. It is composed of polysaccharide, polypeptide, or in some cases it can be made up of proteins and sugars, means both. The glycocalyx may be thick or thin, may be rigid or flexible, depending upon their chemical nature. So the substance which is being secreted, so this, first of all, it may be polysaccharide, it may be proteins or polypeptides. Now, depending upon its chemical nature and texture, it may be rigid, flexible, thick or thin. If it is secreted in more amount, it will be thick. Otherwise, it will be thin. So anything can happen, everything depending upon secretion and the chemical nature. So the two terms are given actually, the capsule and slime layer are frequently used to describe the glycocalyx layers. The rigid layer are organized in the type matrix, is actually called capsule. And if it is layer is more easily deformable and loosely attached to the cell wall, this is actually called slime layer. Most capsules consist of polysaccharides, but this is not 100% true again, because some can also have polypeptides or actually may have both. However, the capsule of some bacilli, for example, Bacillus anthracis, consists of polypeptide and they are made up of D sugars, this is not sugar, this is glutamic acid, this is amino acid. Okay, so normally L amino acids are present, but this is poly-D glutamic acid. Glutamate is an amino acid represented by letter E for glutamate. Aspartate is D. So the possession of capsule makes some pathogenic bacteria resistant to the phagocytosis and enhance their virulence. Okay, so the last line is very, very important actually. The possession of capsule makes some pathogenic bacteria resistant to which? Phagocytosis. Now, who is going to do this phagocytosis? we are having two type of cells in our body. One is called macrophages, one is called neutrophils. Neutrophils are called phagocytic because they are having the similar role as macrophages, but actually they are smaller in size. So, okay, so the actually neutrophils are the first cells. Suppose if some infection happens in our body, some cut or some normal infection, injury. So first cells which are able to arrive to that particular site are called neutrophils. So neutrophils are actually called chote havildar and macrophages are called bade havildars because macrophages are large in size. So macrophages and neutrophils are PMN actually, polymorphoneutrophils. They are having this structure of their nucleus. So depending upon their nuclear structure, they are actually named like PMN. So neutrophils and macrophages are main cells required for phagocytosis. So suppose this macrophage is going to engulf some bacteria, but this capsule or the slime layer or this glycocalyx layer will not allow the macrophage to engulf that particular bacterial cell. So it is making that particular bacteria resistant to the phagocytosis. Okay. And therefore, if the macrophages and PMN are not able to engulf that particular phagocytosize that particular bacterial cell, it is going to cause some disease, and therefore it is making or increasing its virulence. Virulence is a factor to cause a disease. So by a factor, it can cause a disease. It will be increased because it will become resistant to phagocytosis. Plasma membrane, most of you know plasma membrane. So plasma membrane is very similar since it is made up of primary proteins and phospholipids. So in eukaryotes and prokaryotes, almost they are having similar function, but exactly not similar molecular makeup, because one major difference in chemical composition of plasma membrane between eukaryotes and prokaryote cell is that eukaryotes have sterols. So they are having the sterols in their plasma membrane, whereas bacteria generally do not have sterol in their plasma membrane. Okay, so now the question is, if they do not have sterol, then what they have? So they have actually hopanoids. So hopanoids are homologous to the sterols in eukaryotes. Sterols are absent in the plasma membrane of all bacteria except mycoplasma. So there are some bacteria which are very small in size. Actually, these are PPLO, pleuropneumonial organisms. They are having sterols in their plasma membrane because they generally do not have a cell wall. So actually the sterols are, okay, so let me tell you one story first. So we all know that when this earth was formed about 4.5 billion years ago, after about 1 billion year, after about 3.5 billion years ago from today, the first organism which are able to survive the earth were prokaryotes. So the prokaryotes are actually able to means then they evolved and they make different organisms. In fact, the eukaryotes are also evolved from the prokaryotes only. So now, okay, so this eukaryotes and prokaryotes are having some differences. First of all, in their molecular makeup of their plasma membrane only. So actually the prokaryotes generally do not have the sterols. Now came to this specific microorganism, this particular bacteria, class of bacteria, which are smallest known bacteria actually, this mycoplasma. Mycoplasma evolved in such a way that they do not have cell wall. Generally, all the prokaryotes are having cell wall, but these mycobacteria are not having cell wall at all. So now if the mycoplasma do not have a cell wall, it is required that, because otherwise the cell will lyse without cell wall, the plasma membrane without sterols is not able to hold the pressure which is arising due to osmotic pressure inside the cell generating in aqueous environments. Okay, so to stabilize the cell, to stabilize the cell, these mycoplasma along with other eukaryotes actually evolve these sterols. Whereas other bacteria, which is actually present from the initial, is actually having hopanoids, which are homologous to present sterols, which are also present in eukaryotic cell as well as in mycoplasma. Okay, so the sterols are actually having more balancing property of, so that they can stabilize the plasma membrane better because they have evolved from simpler forms from the hopanoid somehow. Okay, so mycoplasma is having sterols because it is not having cell wall, and to survive so that the bacterial cell will not lyse, it will have some more stabilizing lipids, for example, sterols in their plasma membrane. So molecules similar to sterols or hopanoids actually are present in the other bacteria, and many bacteria are likely to play a role, similar role. Now the function is same. Plasma membrane performs many functions, transport, energy, transduction, and so on. There are some structures in the bacteria which are called mesosomes. Mesosomes, we know they are actually homologous to mitochondria. They are for the energy transduction mechanism actually. So an invagination of plasma membrane in the shape of a vesicle, tubule, or some lamellae, actually called this may be in any form, either in vesicle, tubule, or lamellae. So this is called mesosome. Mesosome is present in bacteria that may be involved in, along with this energy role, they also found to be involved in chromosome replication, cross-wall formation. This cross-wall formation actually in dividing bacteria, as we have discussed, MTS-Z. So I need to discuss all these cytoskeletal proteins in the initial classes only, because otherwise you are not able to understand these words like cross-wall formation. Now you are able to understand that this cross-wall formation, okay, so some septum-like structure is formed with the help of some protein which is called FTS-Z, and some other proteins are also involved in attaching this Z ring to the plasma membrane, for example, FTS-A and so on. So actually these mesosomes are also found to be responsible because these are nothing but they are invaginations of plasma membrane. So suppose if plasma membrane is having some invaginations inside, so it is making it, uh, the plasma membrane more accessible to the Z ring, so that they can also involve in the septum formation. Fine. If this is clear or you are having a doubt, this is quite confusing. I understand. If you have a doubt, I will explain it again to all of you. Please ask if you have a query. Actually, mesosomes are present in both gram-positive and gram-negative bacteria, but actually they are more prominent in gram-positive bacteria. So actually they are found in both, but in gram-positive bacteria, they are having more, means large number. Now you can also ask a question why they are more in number in gram-positive bacteria only. Cytoskeletal yes, they are not in Pathfinder. I think they have discussed them in some way, but not in very much details actually. So that's why I finished them and as early as possible. So this should be clear actually. So these mesosomes are present in both gram-positive and gram-negative bacteria, but they are more prevalent in gram-positive bacteria. As you know, the this peptidoglycan synthesis. So cell wall synthesis. So you know, gram-positive bacteria is having more cell wall, thick cell wall, and to make a cell wall, it is requiring more energy. So it will require more mesosomes so that peptidoglycan can be made. Niharika is able to understand, fine. So, okay, so that's all for today. Actually, we will continue our class from this, otherwise it will be mess again. Hope Drishla will not complain again to me that I'm leaving something. So we have to discuss, first we will discuss storage granules. Uh, then we will move to flagella. So we will move in the sequence only. We will skip nothing so that you are able to make an harmony with. So that's all for today. We have discussed some cytoskeletal proteins. Also, we have discussed the outer membrane of bacteria, how this is different from plasma membrane. So also we have discussed the plasma membrane. We have discussed that plasma membrane is having some structures called mesosomes, which are having some role in energy formation, ATP generation, also having some role in chromosome segregation and septum formation. Now it is very clear that how the septum formation is actually happening in bacteria, and this FTS Z ring and FTS A protein is helping the attachment of Z ring to the plasma membrane and so on. So many proteins we have discussed. So I request you please make this table somewhere so that if you try to remind this table 1 or 2 day in continuation, you are able to understand these proteins and their homologs and eukaryotes. That's all for today. Thank you so much for this class. Okay, good night. Have a nice day. Bye-bye.